Good evening, everyone. Welcome to our chat tonight. I see we just got Michael on. Hi, Michael. Once you get on and get situated, the chat box to the right, just a little bit if you could kind of let me know, progress with AP Insight so far. The greatest challenge you're having right now, any tools that you particularly like. And while you guys are doing that,
Hey, Megan.
can you hear me now?
Hey, Megan, can you hear me?
Yeah, Jim, can you hear me?
Can anybody hear me?
What just happened? All right, can everybody hear me right now?
Yes.
Oh, great. Thank you for that. My computer all of a sudden wanted to disconnect my microphone. All right, as you guys are typing in the chat room, hopefully you're finding up in the top hand corner where you can raise your hand. You can agree, you can disagree, but you guys all have microphone rights, so this is supposed to be a chat. So definitely feel free to type in the chat or stop me and get my attention. So I want to welcome you guys all for being here tonight on a Wednesday night at 7 p.m. Appreciate it. So tonight we're going to talk about using AP Insight to enhance the AP Biology laboratory experience. I'm pretty excited about this idea. Audra Ward, who is here with us from the College Board, comes up with some pretty exciting ideas for discussions, and I particularly like this one just about as much as I liked the last one. So the ability to kind of incorporate these things together. So a little bit about myself. My name is Megan Falero. I teach at Durant High School. I am in Tampa, Florida. I have been teaching AP Biology for 8 years. This is my 21st year teaching. And I've been using this program for 6 years. So I'm a big fan of AP Insight and where this program has come over the last 6 years. So I see that we have a pretty wide range of experience. I see some of you that have been using it for a couple of years now. Some of you that are just learning about this program, which I think you're going to love. So tonight, one of the things I'm not going to go too much into detail about what this program is and the nuts and bolts of it, but I do want to talk about how to utilize this program, because one of the things that I always hear a lot when I'm doing these trainings is I hear people saying, it's just one more resource. I already have a million things. How am I going to possibly fit this all in? So I think tonight is a really good way of showing how you can incorporate this program into what you're already doing. And I noticed what Jim said earlier. He said that he utilizes the quizzes, but he adds a little more each year in terms of activities and challenge area assessments. So this is kind of one of these programs where you don't have to use everything. In fact, in the beginning, I probably would encourage you not to try to use everything. So I'm pretty comfortable with this program, so feel free to ask questions as we go. So at this point, if you do have questions about what AP Insight is, on the AP Insight, there is a discussion part. Feel free, I'm the moderator for that site. If you have questions about how to use the program, how to make a quiz, what to do after you give a quiz, that's a great place to ask the kind of questions about the nuts and bolts. But hopefully we can have a nice discussion tonight as well. So where we're going tonight, we want to talk a little bit about the tools and the resources that are on AP Insight that have a particularly beneficial assistance to the lab aspect of our course. So that I'm hoping that in the end we see ways where we can integrate AP Insight into this lab experience so that while we're doing the lab experience, we can use AP Insight for the formative piece or as a piece to support what the kids are getting from their lab. And then hopefully you're going to be able to use that to go back and take the one piece that we're doing, looking at a specific lab tonight, and do that with some of the other labs. I'm very intrigued to do this myself, actually, so that you can use AP Insight and you can start planning instruction and lab experiences that will complement each other. And it is a nice time for us to share best practices and get a chance to talk to each other. So what is AP Insight? I want to mention just kind of where this program has come over the last 6 years. So this program actually started out as a bunch of AP biology teachers, instructors, trying to figure out why AP biology students were one of the least successful AP exams that were out there. And there was a statistic that the number of people getting ones on the AP biology exam outweighed the number of people that were even taking AP physics. So the numbers were pretty rough. And when they started looking at, okay, why is AP biology so difficult to teach and why is AP biology so difficult to master, they started looking at, okay, well, we need this program that would provide some of these problems that people are having. So what AP Insight is it is a bunch of instructional resources. Here in Hillsborough County down in Tampa, when we started piloting this program, one of the things everybody said was, oh, it's going to be a test bank. No, it's not a test bank. There are quizzes and assessments that we can use for formative assessment and feedback, but there are instructional resources. This is not a comprehensive list. You are expected to teach the course, but what they've done is they've looked at the areas of the course that are hardest to teach and the most difficult to learn. So they called those challenge areas for a reason. So it provides focused support for challenge areas. And then they have said that these are foundational, that in our curriculum framework, these are foundational learning objectives to college level achievement that again, teachers are having a hard time teaching and students are having a hard time connecting all the dots. And the idea that we don't have to wait till to July to figure out what our kids know and what they don't know, and adding that formative assessment and adding questions that mirror what we would see on the AP biology exam. So the kids can kind of get used to the language of that style and level of questioning that often my own classroom tests don't get to. So each of the 12 challenge areas for AP biology, if you haven't been on the site, there are 12, and they are arranged by big idea. And in each one, there is this 4-stage process. So in each one, there's a prepare where you can unpack the challenge area. You can figure out what's in it. My favorite part of that is the ability to kind of diagnose student misunderstandings and anticipate where the kids are going to struggle. So even though I've been looking at this site for a while, every once in a while I still pop in to prepare to kind of, okay, reorient myself before I tackle a new topic. In the teach, there are connecting to real world topics. There are performance tasks where the kids are asked to do something or create a model. So these are going to be some of the more instructional resources that you, again, may or may not use. You might have a whole list of things you already do, and you might want to pull just a few of these. In the assess piece, these are where the assessments are. So there are building block quizzes, which are multiple choice assessments that look very similar to the AP exam. And there are challenge area assessments that look like the FRQs. When you're done then, it provides a beautiful class report where you can go ahead and you can look at and diagnose specific misunderstandings that your kids are having. So that's kind of what this program is. It was designed with a lot of research in mind, trying to figure out the best way to support us as teachers. All right, so tonight, kind of want to talk about one particular challenge area. We're going to focus on challenge area 3.11, which is evaluating DNA transmission. And then we are going to look at AP Biology Investigation number 7 specifically. And then we're going to kind of look at how these two can be integrated. And hopefully we're going to have a nice discussion about this so you don't listen to me talk all night. So if we look at challenge area 3.11 and what this actually is, I'll give you a minute to look at this. So as I said, challenge area 3.11, we're looking at evaluating DNA transmission. So this is in big idea number 3. Which is living organisms store, retrieve, transmit, and respond to information essential to life processes. You can see there underneath it the enduring understanding and the essential knowledge statement specifically that were chosen. And then you can also see below that the science practice. So in each one of these challenge areas, just as they are in the learning objectives in our course, science practices are integrated into these learning objectives. So science practice 5.3, the student can evaluate the evidence provided by data sets in relation to a particular scientific question. So we want to make sure that not only are we addressing the content, but we are giving a chance for the kids to practice this scientific practice. So you put those all together and we come up with our first challenge area, which is learning objective 3.11. So that's where I want to make sure that I'm very clear that these challenge areas are a piece of the curriculum, a core piece of the curriculum framework. And a challenge area is a learning objective. So 3.11, the student is able to evaluate evidence provided by data sets to support the claim that heritable information is passed from one generation to another generation through mitosis or meiosis followed by fertilization. So my question to you, I'm going to stop here a minute because I find this challenge area extremely difficult, which is why it's a challenge area. Why do you think that this challenge area is either difficult to learn or you would find this very difficult to teach? Feel free, you can either jump in on the chat box, you can activate your microphone, feel free. I don't think anybody has their sound activated because I can't hear anybody. If you go up, you should be able to click on your microphone and you should be able to select your microphone. I'm glad you guys can hear me. But this isn't going to be a very good discussion if you can only hear me. If you don't have access to a microphone, again, please feel free to type into the chat. Challenge Area 3.11. This is a hard one. I will get the ball rolling by saying mitosis and meiosis. The kids get so bogged down in the details of just trying to understand where the chromosomes are going that they often lose the big picture. So I'm constantly going back to the big picture so that they don't focus on the nitty-gritty details. That's my problem usually. Yes. Oh my gosh. They want to memorize it. Levels of regulation to DNA replication to nuclear division. Hmm. Oh my gosh, yes. Mitosis and meiosis, getting those backwards or mixed up in their head. I try to separate them by a large amount of time actually now, so I can get one cemented in their head before we move on. Oh, that's a really good question. Mhm. Um, so if you look at this challenge area, it is broken up into 4 building blocks. So the idea is that we take that learning objective and then we chunk it into smaller units and we focus on each one of those smaller units. Oh, that's interesting. Evaluate data sets. Thank you for bringing that up, that it might not even be a content problem, but it could be a problem that they can't do the practice asked of them. Hmm. All right, thank you for those comments. So these building blocks, we have building block A, DNA replication, building block B, mitosis, C, we have meiosis, and then we have cell cycle regulation. So let's throw in regulation with all of that. So what we need to do then is we're going to use these building blocks to kind of scaffold the understanding of the content and hopefully scaffold the mastery of the skill. Michael, you bring up an excellent point that they're looking at something that is microscopic, and that often we're showing them still pictures of it, so they don't understand this is a continuous process, and it's hard for them to see. That's a good point. So what we need to do is figure out, okay, if we've got these 4 building blocks, what order are we going to teach them in? And then we need to kind of figure out how they're all going to fit together. So the building blocks are the concepts that the students need to know in order to understand that challenge area. And again, this is arranged and part of the curriculum framework. So we are tackling a big piece of the curriculum framework with this, particularly we're in Big Idea 3. So then when you go under the guide to the challenge area, that prepare tab that I was telling you about, there is a recommended sequence. You don't have to teach it in that sequence. I often change every year seemingly. The way that it's up there on the screen right now is how I did it this year. So I started out with DNA replication, then I went to mitosis, and then we talked about cell cycle regulation, and then moved it into meiosis. So that seemed pretty logical to me. Again, logic always seems to change a little bit each year, so you don't have to do it in that. But the idea is that in order to teach the whole challenge area, you do need to teach all of those pieces of the puzzle. And then at the very end, there is a challenge area assessment where you can see, okay, can they link all four of those building block ideas together? Now that they've chunked the information, got them separately, now we need to link them all together. So one of my favorite, I say this funny, in one of my favorite pieces of AP Insight is in the upper right-hand corner of every challenge area. There are these little concept maps that show what the what the building blocks are, and it shows these dotted lines connecting all of them, showing that you can teach them in any order. The first thing I go to that almost the first keystroke after I click into a challenge area almost every time because I want to get that big picture and try to figure out the order that I want to teach it in that year based off of the strengths and weaknesses of my kids that year. All right, so looking at what that challenge area looks like, then let's go over to laboratory investigation number 7, which is cell division, mitosis, and meiosis. I want to stop here a minute because I'm super curious. How many of you do this laboratory investigation out of the lab manual versus do you use an alternative to this? Is there any other lab investigations that you use that may not be the formal one out of the lab manual? So I'm going to pause here a minute. I'm curious.
Is my speaker working now? Is my microphone working
I
now?
hear you. Yes, I
All
hear you,
right.
Jim.
I do all of it except for the effects of the environment. Um, I have had, and if anybody has any suggestions, I've had zero luck getting students to do the mitosis squash and get more than a couple cells that were recognizable. So I just don't do the squash. I use the prepared slides and do an inquiry thing, but I do the other 4 parts. If anybody has any advice, I'd love it.
So it's actually really, really funny that you would say that because I was lecturing today over plant responses and changes to the environment. We were talking about meristematic tissue. And a whole bunch of kids were like, "Oh, like when we looked at the garlic root tips." And I'm like, "Yes, that was the meristematic tissue." And I said, "So that's why we were looking at that particular area." So they brought it up. They remember making those smashes, and I agree they're not great. So then you go to the prepared slides. But I think just the fact that they made the smash and they were looking at real cells to see that's actually where cell division is happening. I thought that was kind of worth it, and the fact that they brought it up today made me really happy.
Agree. I'm glad you said that because I'll probably do it for that reason. I just wasn't getting the data, but it's the
Yeah,
experience, I
no.
think, is what you're saying.
Yeah,
Thanks.
yeah. We tried really hard to get it to it was thin enough that you can see individual instead of piles of cells, and a couple of them came out decent, but yeah, for them to then go and then compare it to a beautiful prepared slide, that in itself is, it makes them appreciate a prepared slide better.
Right.
Yeah, also the prepared pictures of the Sordaria for sure. I had a dish of Sordaria last year and I'm like, oh, I miss my pictures. Yeah, for sure. So one of the reasons I asked this is because explorebiology.com, Kim Foglia's her lab on mitosis and meiosis, I absolutely think that pieces of that are beautiful. That where you talk about loss of control in the cell cycle with cancer, she has that data set of chicken stomach cells versus cancerous stomach cells, and I just think that's fantastic. So I actually do parts of this, and then I do part of Kim Foglia's lab. So if you are unfamiliar, if you're going, I have no idea what you're talking about, Megan, let's talk just briefly about what this lab is. So part one, no, it's under AP Biology, under the labs. Oh gosh, I'll have to look and see what it's called. But yeah, nope, it's under AP Biology, under labs. It's not in the teacher vault. So part one is on modeling mitosis. So you can use pop beads, you can use socks, sockosomes, or you can use pipe cleaners. This particular year, I was at Target and I saw these giant fuzzy pipe cleaners and had to get them. So I used those in beads that I got at Target, and I had giant whiteboards. So the kids were drawing out cells and moving the chromosomes around on these whiteboards with these giant fuzzy pipe cleaners, and that worked amazing. So just the fact that you are modeling, so here we are, we're using science practice number one is that the students can be able to use representations and models to show scientific practices. So the second part, we are looking at the effects of the environment. So now we're looking at the effect of lectin on cell division in onion root tips. So here, I hear another science practice. So if you're doing part two, the students have to set up a lab. They have to analyze data from an experiment. Part three, well, now we're looking at cell cycle control, specifically with cancer. So now we're looking at karyotypes of a normal karyotype versus a karyotype from Henrietta Lacks, the HeLa cells. With the idea that at the bottom, students also are to complete the case study, but I'm too young, from the National Center for the Case Study Teaching and Science. Again, I don't usually use that case study. There's a whole bunch of them that are really, really good though. Wish I had time to do more. But now we're bringing in, okay, cell cycle control. What if this cycle doesn't have the controls? Part four, then we're back to modeling. So now we're going to model meiosis. So we're back to pop beads, sockosomes, or pipe cleaners. And the students are going to model meiosis. And hopefully changes in the amount of DNA that occur during meiosis. And then stage five, or I'm sorry, part five, we have meiosis and the crossing over. So now we are looking at crossing over frequencies and crossing over in Sardaria. So now we bring in mathematical calculations. So that's science practice number two, using mathematics appropriately. So a lot of science practices in these. So going back to that question that I asked earlier, if we look at what AP Insight has for us and what this lab provides, where are we at? Jim, that's such a good statement. I say that because when I went to the AP reading last year, I was on the mitosis meiosis question. It was one of those 4-point questions, and it basically said two things they have in common, two things that are different. And I thought, wow, this is such an easy question. And it was just unbelievably awful to grade. That was just, kids don't understand mitosis and meiosis. Yeah, good point. Thank you for that. So here are basically three reasons why this challenge area is hard to learn. And I think we've pretty much said all these. Students struggle with mitosis, meiosis, and fertilization. You get fertilization coming into this. They think that meiosis and fertilization, they interchange those. So how does that play? All of those play a role in development, reproduction, evolution. Bridging concepts. Students cannot connect the process of meiosis, DNA replication, and that passing on from one generation to the next. What does that look like? And then the understanding the cell cycle regulation. So all of this that's available on this slide, this is the kind of information that you would find under the Prepare tab. So when you go into this challenge area, you can go under Prepare tab, and this is kind of the information that you would use to plan, going, okay, I already know that this is going to be a struggle, so that you need to specifically address these struggles, as we plan. And then we look at it from the teacher perspective of why is this so unbelievably difficult to teach? And it was brought up earlier of that microscopic nature of that this is a continuous process and that prophase, metaphase, anaphase, these are basically just made up by scientists, but this is one continuous process. But at fixed points, we stop it and say, okay, this is what this is called. Confusion between the similar processes. They get them so confused and backwards in their head. And then going back to that big picture, helping students understand the role that mitosis and meiosis play in that larger picture. So my key takeaway point from this so far is that when I'm looking at these misconceptions, I'm thinking, all right, I know they're going to come, and that I need to make sure that every single time I go to teach one of these things, that's in the back of my mind, and I'm intentionally addressing that misconception with them. And just a little plug for the AP reading. You walk away from that AP reading with a lot of these in your back pocket, if you have not been to the AP readings yet, that you see thousands of examples of what you don't want your kids to say or do. All right, I'm going to pause here a minute. Thoughts, questions? Are you with me? Thanks. Yes, the AP reading is the best professional development you will ever go to. I agree. All right, thanks, guys. Appreciate it. All right. So earlier, it was brought up that it could be just the data analysis piece that kids are having a hard time understanding what to do with data, and that maybe they understand mitosis and meiosis, but they can't evaluate the data piece of it. So this is an example of, in AP Insight, one of the data analysis pieces. It's one of the performance tasks. So when I was talking earlier, under that, you have that prepare tab, you have that teach tab. Underneath that teach tab, there are performance tasks down at the bottom. And these could be like little mini labs. These could be something as simple as asking the kids to make a model, but it is a chance for them to stop. And these are kind of nice because they also have like little check your understanding, check. It is asking the kids to be metacognitive and think, okay, stop. Do you get it? Are you sure? So that's kind of nice. So there is a level of ownership that these tasks bring in. So in this particular performance task, we're looking at the specific building block, we're looking at analyzing DNA, we're looking at Messelson and Stahl's experiment. So we're back to building block A, which is looking at DNA replication and being able to understand it. So this is actually kind of a nice performance task because it actually has you reading, has the students reading the actual original article from Messelson and Stahl, then taking a look at some data and then analyzing it. So that might be a nice piece that after you model mitosis, then you bring this in and you see, okay, do you get it? You understand the chromosomes are separating. Okay, how does that affect the amount of DNA that's available? And then afterwards, because this is building block A on DNA replication, then you can go ahead and you can take a building block quiz. These building block quizzes are 3 to 5 multiple choice questions each. They don't take very long, but again, they're written at a level that is probably higher than what you would write. They're written at the level of the AP exam. So getting the kids used to that wording is super handy. So these performance tasks, they are assessments for learning, and hopefully, we can use those to build understanding. I do some of them. Again, you probably wouldn't do all of them. All right, so if we go back to the idea of addressing all of the student struggles as we go, somebody said earlier in the chat that they teach all of the pieces of this lab, but they teach them separately, which I really, really like. So one of the recommendations that is made here is, okay, AP Biology Investigation 7, you look at parts 1 and 4. So the kids are modeling. So they're on their whiteboards or they have their socosomes or their pop beads. They're visualizing the changes in the amount of DNA. Then they go and they do one of the performance tasks where they have to, in this here, it's molecular variation during the cell cycle. This particular performance task asks them to look at the amount of DNA in mitosis and meiosis, and it asks them to create a visual, create a graph of how would you show that. So it has them really think, okay, you're connecting the amount of DNA to these processes. The next one then is part 2. So then we look at that effect of lectin on cell division. And so where they're performing the experiment, they're collecting data. And then you look at part 3 where they're using the karyotypes and looking at the HeLa chromosomes versus normal chromosomes in a karyotype. And then you can go back to a performance task that's in AP Insight. In this particular one, it's asking them to construct a model of the cell cycle. Interesting. So it's you can see what's kind of even already on this page: construction paper, tape, scissors, markers, whiteboards. Have them create a model to show cell cycle control. Now that we've looked at, okay, when the cell cycle, when we lose control, what happens? So we go back to that, bringing in the science practice, looking at creating a model or a representation. Then the next part is part 5 of the lab where they're looking at counting the crossovers and the sordaria. So after that, once you have done all 4 of these building blocks, the idea is that, okay, you've pretty much covered the whole challenge area. So now can we go back and can we tie all of these ideas together now that they have the little pieces, hopefully at that point, somewhat mastered. So besides the building block quizzes, I also talked about the challenge area assessments, those FRQ style questions. So if you got it out of the reading materials at the bottom of the page in the bottom right hand corner, there is this challenge area assessment if you want to take a look at this, if you went ahead and you downloaded that. Or if you are having another tab open and you're looking at the AP Insight, this is the challenge area assessment for 3.11. So it's under assess, and under the building blocks, there's the challenge area assessments. So if you get a chance, again, I'm going to shut up for a minute, let you look at this. Now that you've covered all of these should be linking all 4 of those pieces. So the kids have to be able to understand mitosis, meiosis, cell cycle regulation, and DNA replication to be able to effectively answer these questions. For those of you that have been using AP Insight for any length of time, I'm curious on Jim's question, ideas on how you utilize these challenge area assessments. Would you use them as summative, formative? Would you use them as homework? Would you give them as FRQs and make the kids write them as in class? This is not a strength of mine. Oh, John, that's a great idea. Using more of them as review because so many of these tie together ideas across the curriculum. using them as review. That's a good idea. In the past, Jim, I've used a couple of them on my unit exams. I will pull them and use them as really a summative assessment. I like using them as a review, though. Could you even use them at the beginning of class as a brainstorming of in there? For me, I have groups. I have groups of three. Could you have the kids outline and make an answer to these? Could these be something to get the kids having a discussion? But if you're looking at these two questions, I'm just looking at the first two for now. 3. You can see how these questions are designed where you are going to know exactly what those student misconceptions are. If there are still any, these questions are going to draw them out. But that's what we want, right? Formative assessment. We want to find out what those misconceptions are so that as readers, we're not reading them in June. So hopefully we can get to the bottom of those. And all of the questions on AP Insight are designed for that purpose. So that if the student, for example, the answer is A, but the student picks answer B, they're very thoughtfully written. So B is this misunderstanding. So if the kid put B, most likely their misunderstanding was ... Okay, so just so you know I'm not lying, here is a question. So this all links back to, because it is in the curriculum framework, Challenge Area 3.11 links back to the AP exam. So this is a question from the 2014 International Exam, and this is a multiple choice question. Give me a minute to look at it. All right. Any questions, thoughts on that one? A good question. And here, same thing, linked to the AP Biology exam, but here is an example of this particular learning objective, but in an FRQ style, which I don't know why I don't give this question. This question is amazing. I'll give you a minute to look at this question.
I can talk faster than I can type. What I'm finding I love about these are there's a lot of in the CAs and in like this question here, kids have to read about a story that they've never seen and then respond to it under time pressure, just like the exam. So I'm realizing that the more I can do that, the better prepared they're going to be under on the test day.
Absolutely. We were talking today with plant responses and plant hormones, and I told him, I said, if they can write a question where they can bring in like 4 completely what you would think are unrelated parts of the curriculum, then it's a good question. So then I had them brainstorming. I said, okay, so plant hormones, what could they ask you about? Could they ask you about biochemistry, polar, non-polar? Can you ask about cell communication? You can talk about ecology and responses to changes in the environment. You can talk about population control. So those are kind of fun though to brainstorm and okay, where's College Board going to take us this year? What are they going to make us try to connect? So this question, this is an amazing question. So you've got mitosis because you've got the cell cycle phases. You've got growth factor. So now you're talking about cell signaling. Um, so you've got a reception, transduction, cellular response that they can bring in. Then you bring in mutations and loss of control of the cell cycle. And then, oh, coding of the region of the gene. Oh my goodness gracious. Oh, how fun is that? And then they have to propose a model. So there's, yep, I agree. There's the modeling again. And a reason why therapy is effective for treating cancer. So this cross-curricular idea, this is a beautiful question. All right, so some questions for you guys. All right, question number 1. Which lab investigations have you found to be the most helpful for you and your students in developing enduring understandings and why? And again, I don't think when I say lab investigations, I'm not talking specifically from the lab manual. It could be a lab that you do that you've pulled from another source.
Megan, I love the artificial selection because number one, I started in September and it's still going on. And like you talked about plants, so as the plants are growing, I get to talk about everything we're studying and applying it to plants. It utilizes all the statistical analysis. It just allows me to connect it to really every aspect of the curriculum. Um, it's there staring at them as the plants are growing, and so it's a great visual cue. But I love that lab for taking them through the whole process of inquiry, plus what it allows me to do content wise.
Hmm. Thank you for that. But now I feel bad because I don't do that lab. But maybe I'll do it now. If you like it that much. It's that good, huh?
Well, it fits into we have the equipment, so it fits in. No, I love it. Yeah, I do. But I'm weird.
Other thoughts? What are your favorite labs? What do you think is really, really helpful? I was just thinking enzymes. I did yeast spheres for the first time this year, and I have to say I'm pretty sure it was my favorite lab this year, where we took the sodium alginate, we made the spheres with the yeast inside of them. Oh, it was so good. So good. It was the best. The kids completely got enzymes this year. Transpiration, absolutely. Very clear cut. My kids, we are in the middle of a transpiration lab right now. Oh, the
The
blast lab.
Carolina enzyme. Oops, sorry.
What
The
Carolina
Carolina
enzyme?
enzyme one. Yeah, the Carolina enzyme with catalase is a nice basic enzyme lab, but then allows you to use a blast type of program like Uniprot and bring in cladograms and comparing amino acid sequences. So it allows us to connect, do what you're saying, connect enduring understanding.
I found this year, and it's different every year, but this year after the blast lab, all my kids were like, I'm not going into bioinformatics. Like, okay, I think I scared them all away from bioinformatics this year. That's a good idea with that transpiration lab. Bringing in osmosis diffusion, you're reviewing from previous concepts, tying things in from earlier, from me, from first semester. All right, question number 2. What have you discovered, if anything, about your students' misunderstanding or your instructional practice through the use of AP Insight or a lab experience? About student misunderstandings or your instructional practice. John, I like that. Different students need to be exposed to different ways of learning. Can you give an example of where you've taught it one way and then you flipped it and used a different learning style to teach it in another way? Metacognition is not a buzzword. I love that word. Okay. Gene to proteins. Okay. So there is a beautiful challenge area for that. So hopefully when you get into that, if you haven't already used the challenge area, um, really good one for that, for transcription, translation. 45 students per class. Oh boy. I appreciate you guys. These are good comments. So I have to brag on something that I saw on the AP Biology Facebook page. I've seen a lot of the stop motion video and using the stop motion studio apps. So for me, cell communication's killer. So I had so many kids that were looking at me like I was crazy when I was talking about G protein coupled receptors and whatnot. So I had to make a stop motion for a cell communication project. They turned out amazing. And my kids got to learn a new technology in the process, bringing technology into the classroom. So I think having to break up labs is, I think in some ways that's better because you can chunk it. And if you look at what we've been doing tonight, where you take little bits and then you work in something else just to make sure, okay, did you get that concept? All right, well, let me see it, model it before you move on to the next part. All right, next question, last question. For the upcoming unit, where do you think incorporating AP Insight resources with the laboratory experience can add the most value for your students? Do you see doing something kind of like what I shared with you tonight, or using a piece of it maybe? Awesome. It's definitely got me thinking, that's for sure. The app for it was Stop Motion Studio. It is a free app. So in all of these challenge area assessments where the kids are creating models, oh my gosh, stop motion, where have you been all my life? Kids had a lot of fun with it too. Oh, the Hardy Weinberg challenge area is fantastic for this. Yeah, I see that working really well to support teaching Hardy Weinberg. Good idea, guys. All right. So if you've got questions for me, um, let me remind you, if you don't know what Share What Works is, it's the last piece of AP Insight that I have not mentioned tonight. This is our online community and discussion board for teachers that are using AP Insight. So most likely when you type into that, you're going to be typing to me because I am the moderator. So please talk to me. This is a chance for you to tell me what's working. I think other people really like to hear it as well, so you can ask questions, certainly, if you just can't figure something out. Share an experience that went really, really well. This is a chance for collaboration because a lot of us, we are usually the only AP Bio teacher at our school, so it is kind of nice to know there are other people out there. So, if you've got any other questions for me, at the bottom, don't forget that there are those reading materials. So if you want to download those, you can use them as a reference at a later date. Also, a recording of this PLC will soon be available on AP Insight. So if you want to go back and look at some of these resources that we've been looking at tonight as well. So before you go, are there any questions? I'm definitely going to be sticking around. I'm not kicking you off, I promise, but I'm also respectful of the fact that it is a Wednesday night and we have school tomorrow. So I'm making the discussion more Kimmy. You can, my suggestion would be that if you're on a particular challenge area, you can subscribe to it and you can get anybody that's typing anything in a particular challenge area that you're using at that particular time. I'm so glad. Yeah, thank you, Audra. Yes, this survey, if you don't mind, before you leave me tonight, if you could take this quick survey to let us know if this was remotely helpful tonight. In the links bottom, so next to the reading material, you don't have to type that whole thing in. You can go down and click the link then for the AP Insight PLC survey. You're very welcome. So while you guys are doing that, I will definitely be around for a few minutes. I really appreciate you guys attending tonight, touching base with me. If you've got questions, I'll stick around, but you know that if you click into the Share What Works, you can talk to me anytime. So thank you for all being here tonight. I'm glad that insight has grown in you. You're very welcome. Yep. I think AP Insight grows on everybody the more you use it. I'm not a facilitator? Audra, I'm not a facilitator? But it is there. Audra said to use the link at the bottom. Yes, you have to follow directions. Let's see. Oh no, I used the link at the bottom and I don't see it. If you go down to, I did the same exact thing. If you go down to the links and click on AP Insight PLC survey, and then goes into the browse to when you click the browse to button and when it pops up, yeah, it's it's different when you go in that way. I had the same issue. Hmm. If I click on the link so it highlights it, then it puts the www.research.net in the bar, and then I just hit the browse to button and it should open up in another window. Yeah, yeah, I tried that. Yeah, if you copy and paste the link, yep, it has different facilitators. Yep. All right. Thank you again for being here tonight. Have a good rest of the week. You are very welcome. I'm glad you guys could be here tonight. Thanks, John. You are welcome, Anne Marie. Thank you. Again, I'm here if you've got questions. I'm not kicking you off, but energizing. I like that. Thank you. Plans for more webinars like this. Definitely there are more plans. Audra is constantly brainstorming really good ideas, so she's good at what I think we need to hear. So yeah, definitely stay tuned. I will be posting it in the Share What Works anytime that a new PLC is coming up. So hopefully I will see you on the next one. Oh, I'm so glad. Thanks. Glad I could help. You guys gave me some great ideas too. I am also energized. Thank you. Appreciate it. Glad we could help. Audra, that's actually a question I'm going to field to you. When the tape for this comes out, where does that go now in AP Insight? It used to go under the professional. I'm not sure where those go now. Christine, I am definitely checking. I'm popping on there real quick to see where they end up. Oh, there they are. Yeah, sorry you missed it too. We had some good discussion, but hopefully you can listen back to it. Okay, I found it. When you go when you go into AP Insight and you hit strategies up at the top, go under strategic planning, and in the first box where it says integrate AP Insight, you can connect via the PLC webinar series. So it's under strategic planning. Oh, and Audra will give you the specific link. Awesome. Thank you, Audra. So I'll be around for a couple more minutes if you guys have questions. If not, after you complete the survey, I hope you have a good rest of the week.